The hum before the name. You're already tired, not in a way you decided, in a way that arrived, the way dark arrives, gradually and then completely. Your eyes have that particular heaviness that is not quite pain and not quite comfort, somewhere in the middle of both, pressing gently from behind. Your body has gone soft against whatever is holding it. Your breathing has slowed to something you are no longer managing. This is not nothing. This specific state, this dissolving at the edges, is the result of thousands of coordinated molecular events that have been running, without your input, since before you were aware enough today to have any input at all. The tiredness you are feeling right now is not a single thing. It is a consequence. Something produced it, the way a room produces warmth when enough people have been sitting in it long enough, and what produced it is still running, still adjusting, still moving things from one place to another inside every cell in your body, including the ones in the muscles that have finally, just now, let go. There is a word for the feeling of being kept running without knowing it. You don't have that word. No one does, really, because the process is too small and too constant to have ever needed a name in ordinary conversation. It only got a name in science, and that name came late, and it came from people who spent years looking at things that were technically too small to be seen, and finding ways to see them anyway. The word, when it comes, will be small for what it describes. Most words are. For now, stay here, in the weight of this. The room around you is probably quiet or close to it, some ambient sound you have already stopped registering, the kind of background that only becomes noticeable if it stops. Your body at rest is still a body at work. That is the thing that takes a moment to settle into, the thing that doesn't quite fit with how rest feels from the inside, because from the inside rest feels like cessation, like the pausing of things, and it is not that. It has never been that. Even in the deepest, stillest sleep your cells have ever managed, the motion continued. Cargo moved, signals went where they were needed. Structures were built and then, when they were no longer needed, quietly taken apart and the pieces used for something else. You have never in your entire life been still at the level that matters most. That is either a strange comfort or a strange unease, depending on how you hold it, and you do not have to decide which one right now. The science of the very small has a particular quality that the science of larger things does not always have. When you study something at the scale of a planet, or a mountain, or even a cell visible under an ordinary microscope, the thing stays where you put it, more or less. It has a shape that persists. It exists in space in a way that feels like the way other things exist in space, occupying it, staying in it, being there when you look again. Go smaller, and that stops being true. At the scale where the most interesting things in your body are happening right now, at the scale of single proteins, of molecules a few nanometers wide, of structures whose size is measured in units that have no everyday referent, nothing to compare them to except other things equally invisible. At that scale, the world does not hold still. It cannot. The thermal noise alone, the constant random jostling of molecules by the heat energy in the surrounding fluid, would make stillness impossible even if stillness were the goal. Everything at that scale is moving, always, whether or not it has anywhere to go. The question is not whether something moves. The question is whether the movement is in a direction, whether it is doing something, whether out of all that noise something has learned, over billions of years of pressure and selection and failure to go somewhere on purpose. Some things have. That is what this is about. That is what is happening inside you right now, in the cells of your retinas reading nothing, in the neurons that are quieting, in the tissues that are doing the slow administrative work of repair that only gets done when you finally stop asking them to do everything else. Something is walking. It has 2 feet, roughly speaking. The feet are not feet in any sense that would be recognizable. They are protein domains, folded structures that bind to a specific surface and release from it in a sequence that produces, at scales this small, something that functions like a step. One attaches, one releases, the first one releases. The first one releases, the second one attaches but further along, and then the first one catches up. And then this happens again and again, in a cycle that continues for as long as the energy holds, and the track remains intact and nothing goes wrong, which is most of the time in a healthy cell, in a body that has been fed and rested and kept within the narrow range of temperature and chemistry that makes all of this possible. The pace of this walking is not slow. At its fastest, this protein can take around 100 steps per second. Each step is 8 nanometers long. If you tried to construct an analogy, if you tried to scale this up to something human-sized, to something that would mean anything to a body lying down in a quiet room, the analogy would break almost immediately because the ratio between this protein's size and its speed of movement has no clean human equivalent. It moves, relative to its own dimensions, faster than almost anything you have ever done, and it does this continuously, and it does this in the dark, and it has been doing this since before you were born, and it will do this until it cannot anymore. You did not ask it to. You did not know it was happening. It did not need you to know. There is a word, somewhere near the end of what you're about to spend time with, that will change the way you hear the tiredness you are feeling right now. Not explain it away. Not resolve it into mechanism and leave it feeling smaller. Something closer to the opposite. The way a piece of music sometimes opens up into something larger than you expected, not because it added more notes, but because the notes it already had turned out to be about something else entirely. They is ahead. For now, the heaviness in your limbs, the slow blink, the way the room has receded to a comfortable distance, the fact that none of this is stillness, not really, not anywhere that counts, just the hum, a road made of itself. It is already being built when you need it. That is the first strange thing, not built in advance, not laid down permanently the way roads are laid down, poured and cured and left to outlast the people who made them, but assembled in the moment of its necessity, from pieces that were already floating nearby, waiting in the cytoplasm like materials stacked loosely at a construction site that has not yet decided what it is building. The track Kinesin walks is called a microtubule, and what it is, before it is anything else, is a tube, a hollow cylinder roughly 25 nanometers in diameter, built from a protein called tubulin that comes in 2 forms, alpha and beta, that lock together end to end into a long chain, and then those chains arrange themselves side by side, 13 of them running in parallel, curving slightly until their edges meet and close into the cylindrical shape that gives the structure its name. The geometry of this is exact. 13 protofilaments, always 13 in most cells, under most conditions. The structure is precise in a way that suggests the precision matters, that the tube would not work the same way if it were 12 or 14. That something about the spacing and the curvature and the surface that results from these particular numbers is what makes the whole system possible. Kinesin's feet fit that surface, specifically, the way a key fits a lock, except that analogy is too static. A lock does not change shape, does not breathe, does not have its own agenda about whether or not to remain a lock. Here is what makes the road strange. It is not permanent. Microtubules grow from one end and shrink from the other, sometimes simultaneously. The end that grows is called the plus end. The end that tends to shrink, or anchor, is the minus end. Tubulin subunits attach at the plus end when conditions are right, when there is enough free tubulin available, when the chemistry of the surrounding fluid favours assembly, and the tube extends, nanometer by nanometer, in whatever direction it happens to be pointing, and then, without warning, it can stop. The plus end can begin to lose subunits faster than it gains them, and the whole structure shortens rapidly, collapsing back toward where it started in a process that takes seconds and undoes minutes of assembly. This is called catastrophe. That is the actual term. The researchers who named it were not being dramatic. They were describing what it looks like. A rapid, total reversal of everything that had just been built, and then, sometimes, it starts again. This cycle, growth, catastrophe, rescue, growth again, is called dynamic instability, and it is not a flaw. The microtubule is not failing when it collapses. The collapse is part of the function. A cell that needed its tracks to be permanent would be a cell that could not reorganize, could not redirect its transport, could not respond to the fact that what needed to go somewhere an hour ago is not what needs to go somewhere now. The impermanence is the point. The road unmakes itself when the road is no longer needed in that direction. Kinesin walks a road that may not exist by the time it finishes walking it. Follow the thread a little further. The direction Kinesin walks is not random. It moves toward the plus end, always toward the plus end. This means Kinesin's directionality is built into the structure of the road itself. The road is polarized, asymmetric, different at one end than the other, and Kinesin reads that difference not as information it receives, but as something encoded in the surface its feet touch. The plus end in most cells points outward, toward the cell membrane, away from the nucleus. So Kinesin, walking toward the plus end, is generally walking outward, carrying things away from the center, delivering to the periphery. There is another motor that goes the other way. Dynein walks toward the minus end, toward the nucleus, carrying things back. The two motors use the same road, going in opposite directions, and the road accommodates both of them without a system for managing the traffic, without signals or lanes or timing, and collisions are rare, and the system mostly works, and no one who studies it is entirely sure why it works as well as it does. That is a detail worth leaving alone for a moment. The tubulin subunits that make up the microtubule each carry a molecule of GTP, guanosine triphosphate, when they are first incorporated into the growing end. GTP tubulin is stable. It holds the structure together. But after the subunit is added, the GTP is hydrolyzed to GDP, and GDP tubulin is less stable, more prone to the conformational change that leads to catastrophe. The growing tip of the microtubule is essentially a cap of GTP tubulin sitting on top of a tube of GDP tubulin, a thin layer of stability balanced on top of a structure that is always, underneath, slightly wanting to come apart. If the growth slows, if the GTP cap thins, if the addition of new subunits falls behind the rate of hydrolysis, the cap disappears, and the GDP tubulin underneath is suddenly exposed, and the tube begins to peel and curl and unravel from the tip. This is what catastrophe looks like at the molecular level, a curl. The protofilaments, released from the tension that held them straight, bend outward at the plus end like the petals of a flower opening in reverse, or not in reverse, because this is not a flower closing. It is a flower made of protein and physics, and it is coming apart, and it will come apart in under a second, and the subunits will float back into the cytoplasm, and somewhere nearby, if conditions are right, a new microtubule will begin. The road unmakes itself, and waits to be needed again. Kinesin does not know any of this. That is not quite the right way to say it. Kinesin does not know anything, in the sense that knowing requires something Kinesin does not have. But the relationship between the motor and its track is not one of awareness. Kinesin does not assess the road before it steps onto it. It does not check whether the plus end is still there. It binds to the microtubule surface because its structure fits that surface, and it walks because walking is what its structure does when the right molecule arrives. And it carries what it carries because something attached the cargo before the walking began. And all of this happens below the level of any process that could be called decision. The road exists, or it doesn't. The motor walks or it stops. The cargo arrives or it doesn't. Most of the time in a healthy cell, in a body keeping itself within the conditions that make this possible, the road exists long enough. The motor walks far enough. The cargo gets there. What happens when it doesn't is a question that belongs to a later chapter. For now, the tube, assembling itself in the dark, reaching outward in a direction that is also somehow inward, into the work of the cell, into the infrastructure of a body that has no idea it is being maintained. Cargo. Something is about to leave. It has been packaged, wrapped in membrane, sealed, labelled in a chemical language that nothing in your body misreads. It sits at the edge of the Golgi apparatus, a stack of flattened membrane sacs near the centre of the cell's sorting facility, the place where things that have been made get prepared for wherever they are going. And it waits, not consciously, not with patience, just in the way that a thing waits when it has no capacity to do anything else until the next part of the process begins. The next part is already beginning. A vesicle buds off from the membrane, spherical, 50 to 100 nanometers across in most cases. Smaller than the microtubule is wide in some configurations, larger in others, always enclosed, always carrying something inside it that the rest of the cell, or the space outside the cell entirely, is going to need. The outer surface of the vesicle has proteins embedded in it, specific proteins, arranged in a pattern that is not decorative but functional, the way a shipping label is functional, the way an address is functional. These proteins are what Kinesin's cargo binding domain will recognize. This is how the motor finds what it is supposed to carry, not by searching, by fitting. The vesicle does not know it is cargo. Orbit the question for a moment. What does it mean for a cell to need something somewhere? The cell is not small in the way a word is small. It is small in the way a city is small when seen from high enough, full of distinct neighbourhoods, distinct functions, distinct populations of molecules that belong in one place and would cause problems in another. A neuron can be a meter long. The cell body where most protein synthesis happens sits at one end. The synaptic terminals where signals need to be released sit at the other. Between them, a meter of axon, a distance that is, at the molecular scale, roughly equivalent to asking someone in one city to send a package to someone in another, except the roads are constantly being built and unbuilt and the trucks are 10 nanometers wide. The cargo has to get there. The signal cannot be released without the proteins that make the release possible. The proteins cannot get there by diffusion. Diffusion across a meter of axon would take longer than the neuron has. Something has to carry them. This is not a convenience. This is the difference between a neuron that works and one that doesn't. There are different kinds of cargo. Vesicles carrying neurotransmitters, vesicles carrying membrane proteins destined for the cell surface, mitochondria, whole organelles, the cell's energy-producing structures, enormous compared to a vesicle, requiring not one kinesin but many working in coordination, their efforts somehow summed into a net movement in one direction. Messenger RNA, in some cases, signaling molecules, the building materials for the synapse itself, shipped from the cell body in packets and assembled on arrival. The cell carries things the way a body carries things, with effort that doesn't register as effort. With a system so practiced it has stopped being visible. You have never felt a vesicle being packaged. You have never noticed a mitochondrion being relocated. The work is happening now in you, under conditions that feel like rest, and it is happening because it has to, because the alternative to this constant interior freight is a cell that runs out of what it needs and then, quietly, stops. Come back to the vesicle at the Golgi. It has been claimed now. A kinesin molecule has bound to proteins on its surface. The motor's two heads are on the microtubule. The cargo hangs below, enclosed, waiting in the way things wait when the waiting is not a state but a position. A thing that is about to move, suspended in the moment before movement begins. What the vesicle is carrying varies. What the system looks like does not vary much. The geometry is consistent. Motor above, track below, cargo suspended between them, and the destination encoded not in the motor structure but in the surface proteins of the cargo itself, in the chemical address that will be read at the other end at the moment of delivery, when the vesicle fuses with its target membrane and releases what it was carrying into the space where it was needed. The motor does not know what it is carrying. The cargo does not know it is being carried. The cell does not know it needs what is coming, and yet the delivery happens most of the time to the right place, at roughly the right time, in quantities that are almost always sufficient. There is no one in charge of this. That is the thing that keeps circling back every time you try to hold the system still long enough to look at it clearly. No coordinator, no signal that says now, just chemistry and structure, and the accumulated result of billions of years of anything that didn't work this way, not surviving long enough to be here. The vesicle is moving now, 8 nanometers at a time, 100 steps per second, roughly, under good conditions. The cargo inside it, whatever it is, neurotransmitter precursors, membrane proteins, something the synapse will need before the next signal fires, does not shift during transport, does not rattle, does not experience the journey in any sense. It simply is in one place, and then, after enough steps, it is in another. The motor walks, the road holds for now, the cargo arrives, or it will. Something in that sentence is not quite finished. The 2 feet. The first head is down. It is bound to the microtubule surface, locked into a specific site on the beta-tubulin subunit, held there by a contact that is not fragile. Not a loose association, not a temporary proximity, but a real structural fit, the kind that requires active chemistry to break. The second head is free. It swings forward, not because anything is pushing it, but because the release of energy from ATP hydrolysis in the bound head has caused a conformational change, a shift in the shape of the motor domain, that throws the free head forward like a pendulum released from a held position, an arc of 8 nanometers through the fluid, landing on the next available binding site along the protofilament. Now the second head is down, the first head is free, the first head swings. This is the step, this is the entire step. 2 heads, alternating, each one spending part of its cycle bound and part of its cycle in motion. The transitions between states driven by the binding and hydrolysis of ATP and the release of ADP and phosphate, each chemical event coupled to a mechanical one, the chemistry and the motion locked together in a sequence so precise that for decades researchers argued about which came first, whether the power stroke preceded ATP hydrolysis or followed it, and the answer turned out to be neither simple nor fully resolved, which is a detail worth keeping. Expand outward for a moment. The neck linker is a short segment of the kinesin molecule that connects each motor head to the stalk, the long coiled coil region that extends upward to where the cargo is attached. When a head binds to the microtubule, an ATP is present, the neck linker of that head docks against the motor domain, straightening and pointing forward toward the plus end, and this docking is what casts the other head forward. The neck linker is not a passive connector. It is load-bearing. It is the part of the molecule that translates chemical energy into directed mechanical work. It is 11 amino acids long. 11 amino acids, a segment of protein smaller than small, doing the work that moves everything. Pull back further, and the motion becomes stranger, not clearer. Because kinesin does not move through empty space. It moves through cytoplasm, which is not a fluid in any sense that the word fluid usually implies. Not water, not even a thick water, but a dense, crowded, almost gel-like environment, packed with other proteins, other structures, other molecules, all in their own constant thermal motion, all colliding with kinesin and the microtubule and the cargo, all exerting forces that are, at this scale, not negligible. The thermal noise that seemed earlier like background interference is here a constant physical pressure. Every step kinesin takes happens inside a storm of molecular collision that would, if the motor's structure were not specifically built to handle it, knock the whole process sideways. It is built to handle it. The binding affinity of each head for the microtubule, the timing of the ATP hydrolysis cycle, the mechanical coupling through the neck linker, these are tuned, through evolution, to function reliably inside the noise rather than despite it. Kinesin does not fight the thermal environment, it uses it. The diffusion of the free head toward its next binding site is not purely mechanical. It is partly thermal, a biased random walk, the noise nudged in one direction by the energy released from ATP, and the combination of directed bias and thermal diffusion is what makes the step work, is what makes it fast enough and reliable enough to function as actual transport rather than as drift. Contract back now. You are lying somewhere soft, or close enough to soft, and your neurons are doing what neurons do in the hours when the body is deciding to rest, which involves, among other things, the continued transport of vesicles down axons that may be a meter long, which involves kinesin taking steps, which involves each step being driven by one molecule of ATP being hydrolyzed, which means that somewhere in the neurons of your brain right now, ATP is being consumed in single molecule increments to move cargo 8 nanometers at a time towards synapses that will need it tomorrow, or in an hour, or whenever the next signal comes. The currency of this process is small, the scale is small, the distance each step covers is small, the aggregate is not small. A single kinesin molecule walking continuously at 100 steps per second covers 800 nanometers per second, nearly a full micrometer. In a neuron a meter long, the trip from cell body to synaptic terminal takes roughly 10 to 20 minutes under good conditions, assuming the motor stays on the track, assuming the ATP holds, assuming no regulatory signal pauses or redirects the transport. In human terms, this is nothing. In cellular terms, this is a long journey, maintained step by step, in the dark, without rest. The step itself takes roughly 8 milliseconds from one bound state to the next, under normal load conditions. Within those 8 milliseconds, ATP binding, a conformational change in the bound head, neck linker docking, the free head swinging forward through 8 nanometers of crowded cytoplasm, the free head landing on the next tubulin binding site, ADP being released from what is now the new bound head, the old bound head releasing, and the cycle resetting, and all of this happening fast enough that 100 of these complete cycles occur every second, and this has been directly measured, with instruments that can detect the motion of a single molecule, that can measure forces in the range of piconewtons, that can watch one kinesin walk in real time and count its steps and feel the tiny force it generates against a held bead. And the number that comes out of these measurements, the force a single kinesin generates, is around 5 to 7 piconewtons. A piconewton is 1/trillionth of a newton. A newton is roughly the force of an apple falling onto a hand from a short distance. A piconewton is so far below that as to be, in human terms, nothing. In cellular terms, it is enough to move a mitochondrion. Come all the way back in. The head is down. A TP arrives. The shape changes. The other head goes. That's it. That is all it is. And then again. And then again. What a TP tastes like. Something has already been spent. The molecule that made the last step possible is gone now. Not destroyed exactly, not annihilated, but changed into something smaller and less useful. ADP and a loose phosphate ion drifting apart in the cytoplasm, no longer capable of driving anything until they are rejoined, recharged, rebuilt into the form that can be spent again. The cell does this constantly. It rebuilds what it uses. It has to. The total amount of ATP in a human cell at any given moment is only enough to sustain cellular activity for a few seconds if it were not being continuously regenerated, and so it is continuously regenerated in mitochondria that are themselves being transported by kinesin along microtubules, a dependency folded inside a dependency, the motor needing the energy that is produced by the organelle. The motor is also responsible for moving. That loop does not resolve cleanly. Let it sit. ATP. Adenosine triphosphate. The name arrives once and then the chapter leaves it behind because the name is not the thing. The thing is the energy stored in the bond between the second and third phosphate groups. A bond that releases approximately 30 kilojoules per mole when it is broken under cellular conditions, a number that is accurate and also meaningless to hold in the body, in the way that all very small or very large numbers eventually become meaningless to hold in the body, felt as size but not as substance. What has substance is the consequence, the step, the shape change, the neck linker docking and the free head swinging and the cargo moving 8 nanometers closer to where it is going. That is what the energy became. Not heat, mostly. Some heat. Always some heat. The cell is warmer than its surroundings for reasons that include this, but mostly motion. Directed motion. Work in the precise thermodynamic sense, meaning energy transferred in a way that produces displacement against a force, meaning something moved that would not have moved without it. The cell does this somewhere in the range of 10 to the 21st power times per second, across all cells, across a human body at rest. The number is not quite imaginable. It layers in anyway, settles on top of what has already settled, the way sediment layers, not displacing what came before but resting on it, adding weight. Here is where I am less certain about how to say this. The relationship between energy and motion at this scale is not like the relationship between fuel and movement at human scale. A car burns fuel and moves. The combustion happens, the heat is converted, the motion results, and the chain of events is long enough and distributed enough that the connection between the energy released and the wheel turning feels like a process, feels like mechanism, feels like something happening over time and space. At the scale of kinesin and ATP, the connection is tighter than that. The binding of a single ATP molecule to the motor domain directly causes the conformational change that moves the free head forward. One molecule, one step. The coupling is almost direct. There is almost no gap between the energy event and the mechanical event. Almost. And the almost matters in ways that I find difficult to hold clearly. The free head does not go precisely where it goes because ATP pushed it there. It goes there because ATP initiated a shape change that biased its diffusion in that direction, and then thermal motion carried it the rest of the way, and then it landed where it landed because that site had the right chemistry for it to bind. The directed part and the random part are both necessary. The energy does not fully determine the outcome. The outcome is a probability that the energy makes very high, high enough to be reliable, high enough that the motor walks rather than wanders, but not a certainty in the way that pressing a button is a certainty. There is something in that which takes time to absorb. The idea that reliability, at this scale, is a statistical property rather than a mechanical one. That the step happens not because it must, but because it almost always does, and almost always has been enough for long enough to build everything. When ATP runs low, the motor slows. Not smoothly. The relationship between ATP concentration and stepping rate follows a curve that flattens at saturation and drops at depletion, and at low concentrations the motor spends more time waiting in the bound state, one head down and the other stalled, unable to complete the step because the molecule that would drive the transition has not arrived yet. The motor is not broken in these moments. It is paused in a specific structural state, waiting. and if ATP does not come, the pause extends, and the cargo does not move, and in a neuron where the cargo was neurotransmitter precursors or ion channel proteins needed at the synapse, the consequences of that pause extend upward through scales until they become a thing the whole body notices. This is how the very small becomes the very large, not through drama, through accumulation, through the quiet failure of a step that was supposed to happen and didn't, repeated enough times, in enough motors, in enough cells, to produce a consequence that has a name at the clinical level, that shows up in a scan or a test or a symptom that a person can describe to a doctor, the entire chain of causation traceable, in principle, back to a motor that waited too long for a molecule that was late, the molecule being late. That is the beginning of some forms of harm. The body you are lying in right now is maintaining ATP concentrations high enough in most of its cells to keep its motors walking. The mitochondria are doing what mitochondria do. The regeneration is running. The steps are happening. And somewhere between the energy being spent and the energy being remade, there is a process so continuous and so automatic that it has never once required your attention, not in any moment of your life, not even now, in whatever hour this is, when your attention has mostly gone somewhere quieter. The spending happens, the rebuilding follows, the motor finds its molecule, mostly. Motion is not the same as direction. A molecule in solution moves constantly. Thermal energy sees to that. It collides, rebounds, drifts, collides again, covering distance continuously without going anywhere in particular, without accumulating displacement in any single direction, without producing at the end of an hour a position meaningfully different from where it started. This is diffusion. It is not nothing. Diffusion moves oxygen from your lungs into your blood. Diffusion moves signals across synaptic gaps. For short distances, diffusion is fast enough. For long distances, it is not. Kinesin goes one way. Toward the plus end. Always. Not sometimes. Not under most conditions. Always, in the sense that the structural asymmetry of the motor and its track together produce a bias so strong that deviation from it is not a behavioral variation, but a malfunction. The directionality is not a preference. It is a geometric consequence of how the motor domain sits on the tubulin surface, of which binding sites are accessible from which positions, of how the neck linker docks and in which direction it points when it docks. The plus end is not a destination Kinesin chooses. It is the only place the stepping mechanism can go. Follow that thread. The microtubule is polarized because tubulin is polarized. Each tubulin dimer, the alpha-beta pair that forms the basic unit, has a defined orientation, alpha subunit on one end, beta on the other. When dimers stack into protofilaments, they all face the same way, alpha to beta in one direction, and when the 13 protofilaments assemble into the tube, they all run parallel, all oriented the same way, so that one end of the tube has beta tubulin exposed and the other end has alpha tubulin exposed. The end with beta tubulin exposed is the plus end. The end with alpha tubulin exposed is the minus end. Kinesin's motor domain binds to beta tubulin. When it steps toward the next beta tubulin in the plus end direction. The chemistry of the binding site and the geometry of the step make any other direction structurally unavailable. One way. Because the road only presents itself one way. The consequences of this extend further than they first appear. In most animal cells, microtubules are organized with their minus ends anchored at a structure near the nucleus called the centrosome, and their plus ends radiating outward toward the cell periphery. This means kinesin, walking toward the plus end, is generally walking away from the nucleus toward the membrane, outward. It is an outbound motor. Things that need to leave the center and reach the edges travel on kinesin. Things that need to return travel on Dynein, which walks toward the minus end, inward. The cell's entire spatial logic, where things are made, where they go, how the inside of the cell maintains its geography, depends on this polarity. The nucleus stays where it is because the motors that anchor it have directionality. The Golgi stays near the center for the same reason. The synaptic terminal gets its supplies because kinesin is outbound and the terminal is at the periphery. None of this organization is enforced by a structure that holds things in place. It is maintained dynamically, continuously, by the sum of all the motor steps happening at any given moment, all biased in their specific directions, all adding up to a spatial order that looks, from the outside, like anatomy. Anatomy as the accumulated result of molecular directionality. The shape of a cell, the shape of a neuron, partly a consequence of which way a motor steps. In neurons, the geometry is more complicated and more consequential. Axons, the long projections that carry signals away from the neuron cell body, have microtubules that are uniformly oriented plus ends pointing away from the cell body toward the synaptic terminal. Dendrites, the shorter projections that receive incoming signals, have mixed orientation, some microtubules pointing each way. This difference in microtubule polarity is part of what makes axons and dendrites functionally different, part of what allows the neuron to send signals in one direction and receive them from many, part of what makes the neuron a directed device rather than a symmetric one. Kinesin reads this polarity, without reading it, without knowing it, in the way that kinesin knows nothing. But the consequence of its directionality, applied to these differently organized tracks, is that different cargoes end up in different compartments of the neuron, sorted not by any active decision, but by the geometry of the roads and the one-wayness of the motors that travel them. Directionality, applied recursively, produces specificity. There is something about a one-way system that is worth sitting with longer than feels necessary. It means the system cannot reverse without a different system. It means errors in delivery cannot be corrected by the same motor that made them. Kinesin cannot go back for something it dropped, cannot return a misdirected vesicle to the Golgi for repackaging, cannot undo a step. What kinesin does in one direction, and correction, when it happens, is someone else's job, a different motor, a different mechanism, a different layer of the system. The one-wayness is a commitment. Every step is. This does not seem like the most stable way to run a cell. And yet, here you are, running. The city at 3 a.m. From far enough away, a city at this hour looks almost still. The lights are on in some windows and not others. Traffic moves but sparsely, headlights tracing slow arcs through intersections that during the day would be impassable. The sounds have thinned to the ones that were always there but buried. A distant engine, something mechanical cycling on and off in a building somewhere. The low-frequency hum of infrastructure that never stops because it cannot afford to. The city is not asleep. It has never been asleep. It has only shed the layer of activity that made the essential activity invisible. A cell, at this hour looks something like that. From whatever distance allows the whole thing to be seen at once, a distance that does not exist in physical space but is available to the mind, there is motion everywhere, constant, uncoordinated in any visible way, and yet not chaotic. Things are moving, things are arriving. The population of molecules inside the cytoplasm is not resting just because the body containing them is trying to. The density of the cytoplasm is something that does not come across in diagrams. Textbook illustrations of the cell interior tend towards spaciousness, organelles floating in a clean fluid, distances between structures rendered at scales that suggest room to move, room to breathe. The reality is closer to a room packed with furniture, every surface occupied, the fluid between structures so full of dissolved and suspended proteins that the concept of empty space inside a cell is almost without referent. Molecular crowding is the technical term, and it is an understatement in the way that calling the ocean damp is an understatement. In this environment, kinesin moves. Other kinesins move, on the same microtubule, in the same direction, sometimes cued behind each other, sometimes passing in ways that require one motor to step around the other, the logistics of which are not fully understood. Dynein moves in the opposite direction on the same track. Accessory proteins called microtubule-associated proteins are bound along the length of the tube, some stabilizing it, some regulating which motors can access which sections, some doing things that are still being worked out. The traffic is real, and it is dense, and it runs without a signal system. The sound of this, if it had a sound, and it doesn't, not one that travels anywhere, would be something like a room full of quiet, independent machinery. Not cacophony, not silence. The sound of things that are built to run and are running. Drift here for a moment. There are roughly 37 trillion cells in a human body. Each one contains, depending on cell type and metabolic state, somewhere between a few and a few thousand kinesin molecules actively engaged in transport at any given moment. The numbers do not simplify when you try to hold them. They just sit there, large and slightly unreal, the way all very large numbers are slightly unreal until they are attached to something that can be felt. What can be felt is this: the motion does not stop, not in a single cell of your body, not right now. Not in the hours when everything above the cellular level has quieted to its nighttime minimum. The transport continues because the cell's needs do not observe a rest cycle. Membranes need to be maintained. Receptors that have been internalized need to be recycled or degraded. Mitochondria drift and fuse and divide in processes that require their relocation along microtubules. Signals that arrive during the day have set off cascades that are still propagating, still requiring the movement of molecules from one compartment to another, still keeping motors walking that might otherwise have paused. The city does not sleep. It changes its traffic. There is a quality to emergent order that is different from designed order, and the difference is audible somehow, felt in the way it sits in the mind. Designed order has a logic you can trace, a blueprint, a plan, a sequence of decisions that produce the outcome. Emergent order has an outcome that looks like it required a plan and did not have one. The sorting of cargo to the right destinations, the maintenance of the cell's internal geography, the coordination of hundreds of motors on dozens of microtubules all running simultaneously. None of this was designed. None of it has a blueprint. It is the output of individual molecules following rules so simple they barely qualify as rules. Bind here, release here, step in this direction. And the aggregate of all that simple behavior, summed across the population, is a system that works with a reliability that would be impressive in something that had been engineered. It was not engineered, it was kept. Everything that didn't work this well is not here anymore. The hum of it, if you could hear it. Not the kinesin specifically, not any one motor, but the whole interior of a single cell. The constant motion, the collisions, the binding events, the releases, the conformational changes rippling through protein structures, the thermal jostling of everything against everything else, the sound of a system at temperature, at work, at the particular kind of rest that is not rest at all, but the continuation of everything necessary at the pace the night allows. You're full of this sound. Every cell in your body is making it right now. You have never heard it, and you will never hear it, and it has been there since before you were aware of anything. Something in that is worth not resolving. When it fails, the first sign is not dramatic. It is a slowing, a slight increase in the time between steps, a pause that runs a fraction longer than it should, cargo that arrives at its destination a little later than the cell's timing expects. Nothing breaks. Nothing announces itself as failure. The system accommodates small deficits the way a city accommodates a delayed truck. Other routes, other deliveries, margins that were built into the schedule without anyone deciding to build them in. For a while the deficit is invisible at every scale above the molecular. Then it is not. Kinesin fails in several ways, and none of them are sudden. The motor domain itself can be damaged. Oxidative stress, the byproduct of the cell's own energy metabolism, can modify amino acids in the motor domain in ways that reduce its affinity for the microtubule or impair the ATP hydrolysis cycle, producing a motor that takes shorter steps, or stalls more frequently, or releases from the track before delivering its cargo. The track can fail. Microtubules that are insufficiently stabilized or that have been modified by post-translational changes that alter the surface chemistry kinesin reads when it steps. The regulatory proteins that control which cargo is loaded onto which motor can malfunction, producing motors that walk without carrying anything, or that carry the wrong thing, or that cannot release their cargo at the destination. The ATP supply can fall for reasons that begin in the mitochondria and propagate outward. The failure modes layer slowly. Over time, that is sometimes years. In neurons, the distance problem makes all of this worse. A motor that fails in a short cell fails locally. The cargo it was carrying covers a small distance. The deficit is small. Other motors compensate. In a neuron with a meter-long axon, a motor that fails early in the journey leaves cargo stranded at a distance from both origin and destination. Other motors must find it, if they can. The cargo waits. The process that depends on that cargo's arrival waits. And in neurons, waiting has consequences that accumulate in ways that are not easily reversed, because neurons are not easily replaced, because the synaptic connections a neuron maintains represent a kind of structural memory that took time to build and cannot be quickly rebuilt if the neuron deteriorates. The deterioration, when it comes, is slow. It arrives the way sediment arrives, layer by layer, each one thin enough to be ignored. The accumulation only visible when enough has settled to change the character of what lies beneath. Alzheimer's disease involves, among many other things, a failure of axonal transport. The tau protein, which normally stabilizes microtubules and helps maintain the tracks that kinesin walks, becomes hyperphosphorylated in Alzheimer's, modified in a way that causes it to detach from microtubules and aggregate into the structures visible in affected brain tissue as neurofibrillary tangles. The microtubules it was stabilizing become less stable, the tracks fragment. Kinesin has less road. Cargo accumulates at the sites of track discontinuity, building up in the axon in clusters that can be seen in imaging as swellings, focal accumulations of vesicles and organelles that were in transit and could not continue. The synapse, not receiving what it needs, begins to lose function. The neuron, unable to maintain its most distant processes, retracts. The connection it maintained is lost. This is not the whole story of Alzheimer's. The disease is more complicated than any single mechanism, but this thread runs through it. The failure of the roads and the motors and the delivery system and the slow starvation of synapses that were supposed to last a lifetime. Parkinson's disease touches a different part of the same system. Mutations in a gene called LRRK2, one of the more common genetic risk factors for Parkinson's, produce a kinase that can phosphorylate kinesin-binding proteins in ways that impair the motor's ability to attach to its cargo. Motors walk. Cargo stays behind. The specific neurons affected, the dopaminergic neurons of the substantia nigra, are among the longest and most metabolically demanding in the brain, and they depend on axonal transport with a dependency that shorter neurons do not have. And when the transport fails in them, the failure produces over years the movement symptoms that define the disease. The motor walks, the cargo stays, a step that completed but accomplished nothing. There are other diseases in this space: hereditary spastic paraplegia, Charcot-Marie tooth disease, certain forms of motor neuron disease, all involving mutations in kinesin genes or in genes encoding proteins that interact with kinesin, all producing their symptoms through the same basic mechanism: cells that need to maintain structures at great distances from their nuclei, dependent on a transport system that has been quietly compromised, slowly losing the ability to sustain what they were built to sustain. The failure is molecular. The consequence is a person. That chain of scale, from a single amino acid change in a motor domain to a clinical symptom a neurologist can observe, is one of the longer chains in biology, and it is real, and it is worth holding for a moment without trying to resolve it into something more comfortable. The motor is small. What it carries matters enormously. The distance it has to cover in some cells is unforgiving. Most of the time it makes it. Family. Two motors are on the same microtubule and they are not the same motor. They share a shape, roughly, the way siblings share a shape, the resemblance legible but not complete. The difference is more interesting than a surface comparison would suggest. Both have two heads, both walk on microtubules, both use ATP, but one of them moves fast and the other moves slow, and one of them releases its cargo at the cell periphery, and the other holds on past that point. And one of them is found in nearly every cell type, and the other appears mainly in neurons, in the specific context of mitosis or meiosis, or something more specialized still. Same family, different jobs, different histories, written into their structures in ways that only become visible when you look at the amino acid sequences and find the conserved core, the motor domain, the part that has not changed much in 1 billion years, surrounded by regions that have diverged in ways that reflect 1 billion years of different pressures in different contexts. The Kinesin family has 14 subfamilies, by the current reckoning. Kinesin 1 through Kinesin 14, each defined by the sequence of its motor domain, each doing something that the others either cannot do or do differently. Kinesin 1 is the one this chapter has been circling. It is the conventional kinesin, the one identified first, the one whose structure has been most thoroughly mapped, the one whose stepping mechanism has been watched in real time with optical tweezers and fluorescent labels, the one whose failure modes connect most directly to the diseases named in the chapter before this one. When someone says kinesin without qualification, they usually mean Kinesin 1. The way someone saying car usually means a specific kind of car without specifying it, because it is the most common referent. But Kinesin 2 exists, and it is different. It has 3 subunits instead of 2, a heterodimer of motor subunits plus a non-motor accessory subunit called KAP, and it moves differently, and it specializes in the transport of cargo along the axoneme, the microtubule-based core of cilia and flagella. Without Kinesin 2, cilia cannot be built or maintained. Without cilia, certain cells cannot sense their environment, certain organs cannot move fluid across their surfaces, certain embryonic processes that require the directional movement of signaling molecules go wrong in ways that affect the left-right asymmetry of the developing body. One family, one member's absence, the heart on the wrong side. Orbit for a moment around what it means to be related at this scale. The motor domain, the part of the kinesin that actually binds to microtubules and generates force, is the conserved core. Across all 14 subfamilies, across species as different as yeast and humans and plants, the motor domain retains enough structural similarity that you can look at the sequence of an unknown protein and recognize it as a kinesin by its motor domain alone. This core has been maintained through all the divergence and specialization because it works, because what it does is so fundamental to the cell's needs that any mutation that significantly altered its function was lethal, was removed from the population before it could be passed on, was selected against so consistently for so long that the core has become nearly invariant. The rest of the molecule is where the variation lives, the stalk region, the cargo binding domain, the regulatory sequences that determine when the motor is active and when it is folded into an inactive conformation, the accessory subunits that mediate cargo attachment. These are where the subfamilies diverge, where 1 billion years of different cellular needs have written different solutions onto the same basic platform. The motor domain is the sentence structure. Everything else is what the sentence is saying. Kinesin 5 works differently from all of them. It does not carry cargo. It has 4 motor domains, not 2. A homotetramer, 2 heads pointing in one direction and 2 pointing in the other, and it functions as a molecular cross-linker between anti-parallel microtubules, walking on both simultaneously, pushing them apart. It is a spacer, an organizer, a force-generating element that helps separate the chromosomes during cell division by sliding the microtubule arrays of the mitotic spindle apart. Without Kinesin 5, cells in many organisms cannot complete mitosis. Kinesin 5 is the target of a class of cancer drugs. Inhibiting it prevents dividing cells from completing division, which is, in the specific context of tumour cells dividing faster than they should, a useful property. The same motor family. One member moves cargo, another member moves chromosomes. A third maintains cilia. A fourth regulates the length of the mitotic spindle by controlling how fast microtubules grow. The motor domain held constant, everything else negotiable. What I find strange about this, arriving at it now, is the relationship between conservation and variation that it implies. The idea that evolution can hold one thing nearly fixed for 1 billion years, while allowing everything around it to diversify into 14 distinct functional roles, that the core can be stable while the periphery explores, that the same stepping mechanism can be recruited for transport, for chromosome segregation, for cilia maintenance, for spindle organization, just by changing what is attached to it and when it is allowed to run. It suggests that the stepping is the hard part, that once evolution found a way to turn ATP hydrolysis into directed mechanical steps on a polarized track, the question of what to do with that capability was a secondary problem, one with many solutions, one that the rest of the molecule could be adjusted to solve in different ways for different contexts. The step is old. What carries is newer. What the cargo is, newer still. Something in that sequence, the deep age of the mechanism and the relative youth of its applications, does not quite finish resolving when you first look at it. It is still sitting somewhere, probably looking for where to land. The listener's own interior. You're at the edge of sleep, or already past it, or somewhere in the corridor between the two that has no clean name. The place where sounds still arrive but meaning has stopped attaching to them reliably, where the body has gone heavier than it was an hour ago, and the mind has loosened its grip on the particular shape of the day and let it start to blur. In the cells of your retinas, if your eyes are open, kinesin is walking. In the neurons of your visual cortex processing whatever dim signal your eyes are sending, kinesin is walking. In the dopaminergic neurons of your substantia nigra, which are among the longest and most metabolically expensive neurons in your brain, and which will be among the first to show the effects. If the transport system ever significantly falters, kinesin is walking. In your fingertips, wherever they are resting, against a screen, a pillow, a blanket that has been warmed by proximity to your body, in every cell of every fingertip, kinesin is walking. It has been walking since before you were born. It walked in the cells of the embryo you were, when the cells were still figuring out what they were going to be, when the microtubules of the mitotic spindle were being organized and the chromosomes were being sorted by motors in the kinesin family that have been doing that job in cells very similar to yours for hundreds of millions of years. It will walk in your cells tomorrow, and in a year, and in whatever long future your body manages, the pace of it adjusting with your metabolism, the cargo changing with your cellular needs, the track being built and unbuilt and rebuilt as your cells respond to whatever you're doing and wherever you are doing it. The weight you feel right now, the particular pull of gravity on a body that has finally agreed to stop moving, is partly the consequence of this, not directly, not simply, but the signals that produced the tiredness you felt an hour ago, that initiated the cascade of hormonal and neurological events that brought you here, to this position, at this hour. Those signals travelled as chemical cargo, moved by motors along tracks, delivered to receptors that changed their shape upon receiving them and initiated the next step in the chain. The sleepiness arrived the way all chemical signals arrive, carried. Melatonin was synthesized in your pineal gland and released into your bloodstream, but before it was released, the enzymes that produced it were transported to the right cellular compartments by motors on microtubules. The adenosine that accumulated in your brain during the hours of wakefulness and signaled the growing need for sleep, the receptors that sensed it and changed the firing patterns of neurons in your brainstem were maintained by ongoing axonal transport. The machinery of sleep onset depends, several steps back from the sensation, on the same walking that is happening now inside you in the dark. You did not feel any of this. You felt tired. There is a distance between the molecular and the felt that biology has not closed and probably cannot close, not because the connection is not real. It is completely real, fully traceable in principle, the chain of causation unbroken from the stepping of a single motor domain to the experience of a heavy body at rest. But because the levels between them are so many, and the emergent properties at each level so different from the properties of the level below, that no description of the mechanism fully accounts for the texture of the experience. The tiredness is real, the kinesin is real, the connection is real, the distance between them is also real. This is not a problem to solve. Somewhere in the axons of your brain, cargo is moving. Somewhere a vesicle is 8 nanometers closer to where it needs to be than it was a moment ago. Somewhere a motor has taken a step that it will never take again. Not that specific step with that specific ATP molecule on that specific tubulin binding site. And the step is gone, spent, complete. Its energy distributed into the motion of the cargo and the heat of the cytoplasm and the cellular economy that will use what it can and release the rest. The step happened. The cargo moved. The cell is, in some measure too small to name, different from what it was before. This is happening now, in you. At the scale where the difference between living and not living is maintained, step by step, in the dark, without your knowledge or your help, by something too small to see that has been doing this since before eyes existed to see anything at all. The hum is still there. It was always still there. It will be there when you wake, and it will be there as you cross into whatever the other side of this hour is, and it will be there in the morning when the room is bright, and the molecular machinery of your cells shifts its cargo to match the demands of a body that is, once again, awake and moving and asking things of itself. Now the step takes. Now the cargo goes. Now the motor finds its molecule and the neck linker docks and the free head swings forward 8 nanometers through the crowded dark of a cell that does not know it is inside someone who is almost asleep.